We are moving and grooving. Yes. I have a child in here, so let's see what happens with this episode. We'll make it go. We'll make it go. We'll make it go. Are we all ready for a show? It is live. We are live. We are here. You are here. And we are all ready to rumble with the science news of the week. Starting in 3, 2. This is TWIS. This Week in Science, episode number 720, recorded on Wednesday, May 8th, 2019. No, I have the wrong thing up. Never mind. Hello. I can't do the intro. I got to do it again.
What year did you say it was?
Wait, 2019.
Is it really?
Mm-hmm.
Oh, my goodness. Blair, did you know this?
I sure did.
I had no idea it was 2019. That's, I'm making small talk while you find the right thing.
I got the right thing.
Oh, okay. Never mind.
Okay. Starting again. In. Oh, wait, I need to open iTunes.
Really? I could have sworn just like, I don't know, a couple months ago, I was walking around. It was 2018.
We're almost halfway through already.
Is it halfway almost? My goodness, how time flies. Time flies like an arrow. Fruit flies, on the other hand, they like a banana.
I like bananas. Starting in 3, 2. This is TWIS. This Week in Science, episode number 720, recorded May 8th, 2019. How to Stop the Extinctions. Hey everyone, I'm Dr. Kiki, and tonight on the show, we are going to fill your head with phages, fly sex, and hot ice. But first,
disclaimer. Okay, when the world around you seems to have lost its mind, when up is down, left is right, in is out, and forward is a bowl of tuna salad, it doesn't mean that you are wrong, despite the fact that those around you might think that you are. A recent Makety-Up poll, which despite being Makety-Up, did include a data set of all humans, found the average human only to be of average intelligence, capable of average insight. They were also of average height, weight, and had average inseams, made average points and counterpoints in conversation, experienced just the average levels of serotonin when hugged, and most strikingly, had only an average knowledge of science. And while the average person may think that you are wrong about something, keep in mind that they have a below average understanding of what is going on in your mind. Because your mind is an outlier. You have beaten the averages, enough so as to make the average analyst question your data point. And you have done so in the most extraordinary way imaginable by tuning into This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are, yes, back to talk about science because you know how much we love it. We love bringing the science every week. This weekend, I get to go to a Portland event called Science Fest.
Nice.
Yeah, it's a March for Science Portland organized event. Not marching, we're celebrating. We got science going on. So it's very exciting. It's great to celebrate science. And I love the fact that we also do it every week here. It's a celebration every single week of all the awesome discoveries and new questions that we can ask. Well, let's see. What kind of questions have people been answering recently? I have stories about hot ice, heavenly stars, and hearts. Justin, what do you have?
Let's see. I've got the new frontier in the modern phage of antibiotics. How having room to breathe is a good thing. And finally, human evolution story I don't like. Oh, and just under the wire, a lost hallucinogenic stash from 1,000 years ago.
I'm glad we're getting that story in. Blair, what is in the animal corner?
Oh my goodness. I have the aforementioned fly sex. I have some very thinky wasps and pandas.
And pandas. And we know it must be a good story considering how much you love the panda.
Begrudgingly brought the panda story. I am still a scientist. I am still trying to be fair and balanced. It is interesting science, so you'll hear
This
it.
weekend, begrudgingly do something information I didn't want to agree with. Love it.
All right. So since the major media, for the most part, decided that they weren't going to talk about this story very much, we're going to put it at the very top of our show. Hey, over 1 million species are going extinct.
Woo! Wait, why are we celebrating? Hang on. It sounds like a
It's
bad
happening.
thing.
Oh, it's not a bad thing. Oh, yeah. And as we jump into the show, if you want to hear about this story and you want friends to hear about this story, subscribe to the podcast. You can find information at twist.org. We're also on YouTube. Okay, so this looming extinction, over 1 million species are threatened thanks to people and what we are doing to the planet. A study, a massive analysis, meta-analysis. We've talked about those on the show before. Meta-analysis is when you take studies and you analyze, lump them all together. So essentially you put all the data together, even though the data has come from different places, and you combine them into one overarching study to come to conclusions. And these are the kinds of studies that really give us an idea of what a field of science is saying about something, how the results are all working together or not. 15,000 studies looked at since about the 1950s or within the last 50 years.
That's a lot of studies.
Yeah,
a lot of reading.
Yeah, it's how did you do this? It's got to be computers involved.
Computers,
in
for
this.
sure, computers, yeah.
A lot of reading. Oh,
Either that or a lot of interns.
Control F.
And so these studies were analyzed, looking at research from over the last 50 years, looking at biodiversity, climate, ecosystem health, all sorts of things. And in that time, our human population has gone from 3.7 billion individuals to over 7.6 billion individuals. And because of human activities, species are starting to vanish. And what it amounts to from this study is that 1 in every 8 animal or plant species on Earth are under threat.
Is this including the ones that have already gone since humans made their rise, or is this is current species that are alive?
Species that are alive now.
Oof. That's not good.
No, these are species living now. Some of them are already very well threatened and on the endangered species list. Others are just waiting to die because of things that people are doing. This study was published on May 6th. It is an intergovernmental science policy platform on biodiversity and ecosystems services publication. This is a group that has 132 nations involved. And the full report will be released in about 6 months. But this report that they released prematurely is just a preview of what's going to come. More than 40% of amphibian species are threatened, 33% of marine mammals, 33% of sharks and reef-building corals, 10% of insects.
That's where all the oxygen comes from.
Yeah, many of these species, there are many that may vanish within the next few decades. So not all of this is something that'll happen in 100, maybe 200 years. down the road, but actually stuff that could happen very soon. Now, the reasons that this is happening is human activity, climate change, our use of land, our agricultural practices, many ways. And so there are 5 ways that an article in Science News online, you can find it at sciencenews.org, they have come up with the things that are really speeding up all these losses. And these are our land use. So about 75% of land on Earth has been altered by humans, severely altered by humans. And this means that we have things that seem good on the surface are not good when you really start to look at it. For instance, we decided that we want to use more biofuels. But instead of relying on waste materials for biofuels, there is land clearing for the increased growth of palm oil that can be used as a biofuel. And so that's just one example of the many ways that things that look good on the surface are being implemented poorly and without much thought to how they can be done in a sustainable manner. Also, overfishing in the oceans. And along the lines of what's happening in the oceans, there is another study out this week in Nature as well, reporting on phytoplankton in the subarctic Atlantic area. Researchers using satellite data have been able to create a marine productivity proxy to determine how plankton, really these phytoplankton, are producing in the Atlantic, in the subarctic Atlantic area. And really they find that in the industrial era that productivity is on the decline, and it might be evidence of a predicted collapse of northern Atlantic planktonic stocks.
It doesn't sound good. That's like one of the keystones. Look at the part of the food chain where everything else eats, and then the fish get bigger, the creatures get bigger as you go up the food chain. So then everything relies. Oh gosh.
Yeah, it's like if all of a sudden all the trees were gone.
Yeah. What will we do then? And then on top of that, we're not working on climate change fast enough. We've only got 12, 13 years to really step on the gas. We can do it. We really can, but we're not tackling it quickly enough. And the warming that has already occurred is part of what they think is behind that phytoplankton decline and other effects. Also, we're polluting. We're still polluting. I mean, there was a report out today or this week that I saw that people go to festivals. If festival season is upon us, you want to go see your favorite bands, you want to camp out for a long weekend or whatever, and you want to hear the music. People are buying tents that are being sold as single-use tents.
Oh my God.
Each of these tents, and then they leave them. They get destroyed in the weather at the location, wherever it is. Each of these tents that gets used once and thrown away after one of these festival events is the equivalent amount of plastic to something like over 9,000 straws.
Wait. Okay.
So we're worried about straws, and yet people are buying tents and throwing them away after one weekend. So we are polluting, we are wasting, we are not making changes and choices the way that would be a better way to live. And all of this is leading to also moving of invasive species from place to place as well. So we can change things. We can change it. We just have to start making different choices, everyone. It's time. Okay? So I'm just starting out the show on the message. Let's change things. We can do it.
Yeah, okay. Real quick, I think that one thing that's jumping out at me from this right away is that a bunch of those animal groups that you talked about at the beginning that are under fire right now, they all have one thing in common, and that's living in or around water. Amphibians, sharks and rays, corals, right? But also this idea of climate change, pollution, they affect the ocean hardcore. And I think something that people that are living in metropolitan areas or landlocked areas, they forget that we are all connected to the ocean. And the ocean is a really important biome for us to consider when we think about conservation and saving species and saving the planet as a whole. And as opposed to it being this kind of out there, deep down under the water conversation, elevating that part of the conversation could potentially do a lot to help all of the plants and animals on the planet, because the ocean also greatly affects climate change directly.
Yes. It's all connected, everyone.
connected.
It really is. We live in a massively connected biosphere Earth, and our choices affect people and animals on all around the planet. And in terms of this report that has come out, I mean, yeah, okay, you might go, okay, these species are under threat. What's it to me? Why do I need to make these changes? If the species die, then it will affect our ability to survive on the planet as well. So, this is something that we should take to heart and think about how to make a better future for humans.
For
all
us,
of
for the
us.
next generation, for the
Yeah.
generation after.
And we can do that. We can do it, everyone.
We could, but
And-
here's the thing. Okay.
Oh no, I was going to move to my next story, but
I mean, part of it in a way is I always feel like a little bit of victim blaming. I mean,
mhm.
the average consumer doesn't often have a choice that is sustainable. So
We can vote.
We can vote. We need to do
vote.
things like that. We need to push the
Mhm.
policy side of this to make big changes across industry, not just our individual efforts. Because that alone actually is definitely not enough.
Yeah, the easy choice needs to be the good one.
We can
And
do the thoughtfulness.
That's yes, it needs to be the top level decision.
Yes.
Absolutely.
And maybe don't buy that single-use tent for that festival this summer.
That is
seems
shocking
like a
and
bad
upsetting.
idea. That
For
seems like a
sure.
really, that's the thoughtless thing. But that's not gonna. It's not quite gonna do. But here's the
better
thing.
choices there.
We spend more on pets, pet food, pet toys, sending our pets to the doctor than we spend on science in this country. So we really do need a priority shift. Not saying get rid of your pets, but it's probably we should be putting our resources to creating an environment where our pets will be able to have future generations of pets.
Yeah, everybody, people love their fur babies. Let's make a better future for our fur babies. Ha! Okay, going from Earth, let's travel out into the depths of space. And when we look around the universe, we're looking for signs of water because water could be life. And here on Earth, we look at water to try and figure out what different forms it may hold. How would water behave in different situations? We traveled past Neptune and Jupiter, the Saturn, these big gas giants, right? We've traveled past the gas giants. And if we go based on our own assumptions of our magnetic fields from our own planet, you would think that, okay, they're going to have like a bar magnet, north and south pole kind of magnetic field going on. We traveled past these gas giant planets, which we assume are, gassy on the outside, and then pressure as there's more and more material going into the center of the planets, pressure builds up, and so that gas gets pushed into different phases and into different forms. And so eventually you end up with something. People have hypothesized that maybe there's some kind of a liquid and maybe even a liquid metal interior to these gas giants. But we've never been able to really figure that out. We flew past them and measured their magnetic fields, and they kind of had these weird, lumpy, dispersed magnetic fields that weren't anything like the typical north-south bar magnet that we are looking for. And so we're like, what is going on there? Go back to our search for water around the universe. Researchers have been trying to figure out, okay, let's look at what water is doing. And there have been some researchers working at Lawrence Livermore National Laboratory in California and also at the Laboratory for Laser Energetics in Brighton, New York. And these researchers were like, okay, let's see what we can do to put water under more and more pressure. And maybe that'll tell us what water would be doing if it's in one of these gas giants. If water is in the gases and it's getting pressed in there, what would the water be doing and how would it form up? So at Lawrence Livermore Laboratory, they've been using lasers really strong lasers to pew, pew, right at water to induce shock waves in the water that would synthesize an increase in pressure, to make it seem as if there was increase in pressure going on. And they've been able to see these amazing different forms of ice. We've seen ice, and they have these forms of water ice where ice, the water molecules, H2O, they bond together in these crystalline lattices, right? Increase in ice, decrease in temperature, decrease in movement between the molecules. They're forming up into a crystal. And under different amounts of pressure, they've been able to find different forms of ice. And so we've got ice 1, ice 2, H3, 4, all the way up through. There's an ice 9, like the one in Cat's Cradle, but it's not like the one in Cat's Cradle. It's not a doomsday. It's not going to destroy the world. But now, and recently, they were able to get up to ice. They were able, at Lawrence Livermore National Laboratory, they were able to get up to ice 7. And that was something that they could see. It was cubic, crystalline. You could see it through it like ice. It was transparent to the naked eye. And they were able to super pressurize the ice at around 4,700 degrees Celsius. It conducted electricity, and there were charged protons moving around. And so all these properties, they're like, oh, this is really cool. But we need to look at stuff with X-ray diffraction to really see whether or not there's a crystal forming, because hypothetically there's a crystal there, but we don't know that for sure. So we need to use X-rays, and we need to see the crystalline structure, and we need to make this happen. So they moved on forward, and they used lasers at this New York laser facility, and pew-pewed a laser at a drop of water at such an intensity that it increased the pressure and the temperature of that water to a point that they created ice. I think they did. What is the new number? They called it. No, it's like ice 13.
Wow, they jumped a bunch of them. Wow.
It's no, actually, I'm sorry. It's now. I'm very sorry. Not ice 13. Roman numerals. I missed a numeral there. It's ice 18.
18. They doubled the number of ices. I don't. Yeah. Okay.
Yeah, this is the name for it.
Right.
And if you don't want to call it ice 18, you can also call it super ionic ice.
Better, much better.
That one.
Much better. This super ionic ice is linked together in a completely different crystalline lattice than we have ever seen before. The molecule under this pressure has entirely broken apart. The oxygen and the hydrogens in the H2O have gone, forget it. And where normally the hydrogens bond, and all the oxygens are bonded to each other in a crystalline lattice in this situation. It's an oxygen-based crystalline lattice that makes it very, very strong, so that the likelihood of it melting is very slim. It's incredibly dense. So it's about 4 times the density of your normal water. So you take your normal gallon of water and multiply it by 4. It's going to be very heavy. You would probably not want to pick it up.
But usually ice is less dense than water, right?
Yes.
Yeah.
Right.
So
it's less dense.
So this went the opposite way.
It's the opposite direction. It's incredibly dense. And that density, it's also at an incredibly high temperature. We're looking at temperatures in the thousands of degrees Celsius. It's a very hot ice. This is ice that is not what you find in your freezer. It's ice at thousands of degrees. And because the oxygens are bound to each other so strongly, the hydrogens kind of move around, and the hydrogen movement makes it almost like a liquid. And people are saying that this is not just a new phase of water, but possibly a completely new state of matter that has never
What
been
on
seen
earth?
And
Water science.
Water
You
science.
thought the folks studying water science Really, what could they possibly come up with?
What they
Why
let
can't they
me
buy
tell
the-
you,
They have
it's
2
pretty
atoms
amazing.
or 3 atoms. It's but only 2 elements. There's not can't be that much to discover. Just can't
So
be.
you're telling me
matter.
high school
Wow.
students now, when they learn about phases of matter, are going to have to learn about super ionic forms of matter?
Probably not high school students, maybe college students. Yeah.
Okay.
It's
Well,
new.
in the far
It's
distant
different.
future, it'll be
It
grade
was-
school kids. But that's
it-
okay.
Yeah.
This form of water was hypothesized about 30 years ago, but we did not have the technology until now to be able to see its structure and to make it happen. It's only the advances in laser technology that have happened even just within the last 5 years have allowed this to occur. And so this laser, pew pew, shock waving the water, and then they shot x-rays at it at the very exact second that laser hit it, and they were able to illuminate the formation of these crystals and see what happened to this water at such a high pressure and temperature that it is something that is completely new. And this something that is completely new, what did I already say? It's really dense, it's hot, and it's black. Where other forms of ice have been transparent or you can see through them, no, the density, the way it's linked, it absorbs light.
Wouldn't that
How?
would that make it super ironic ice?
Ah,
Ah,
maybe.
Angsty.
And
I don't.
yeah,
Oh.
and then we move on from here. Let's go back to our planets. Gas giants. They are plentiful around the universe. And from what we are seeing, if they
I
if
forgot where
this
this story
water ice,
started. Yeah.
we're looking at
it's
the
huge.
universe.
Yeah.
That's huge, possibly containing lots and lots of water. And so the researchers think that this water ice form, that in a gas giant, what you have is the gas around the outside, and then it gets a more liquidy form. And then you have a mantle, a thick mantle of this solid liquid ice that has slight electricity flow through it, which would possibly explain the weird magnetic fields that we've measured from these gas giants. And if we take it from there, it could also mean that this form of water is the most predominant form of water in the universe.
Not only that, but because it's in such a dense state, the amount of water that is available in the solar system or in the universe is phenomenally more than we expected. Could even have been out there with every planet covered in an ocean. It would probably not be as much water.
Right.
So
Exciting.
that means it's water is a plentiful thing throughout the universe.
Yeah.
Wow.
So the question now is, this is an experiment here on our planet where they're looking at pure water. What is the situation in a gas giant? What happens to the water as if it's mixed with organic molecules, for instance, methane, ethane? What happens? Can you have a superionic form of ammonia? Because that's one that has been suggested as well. So there are questions that still need to be answered, but it's very fascinating.
Quick question, one you might not have the answer to. So it's not just heat, but it's also pressure that creates this. So is this an amount of pressure that we know exists on these gas giants?
Absolutely.
Yeah, and that's where they went. I think they back engineered it based on.
I just felt like they were cranking it as far as they could go to see what would happen.
So yeah, but there's plenty of different gas giants out there.
All right, fair enough.
It's a bigger one. Yeah, so you're right. But the other thing is, then I wonder, like, what are the possibilities of escape for this water? Is there some sort of would it have to be another planetary impact of some sort? How would you possibly get this water removed from one of these cores so as to sprinkle it liberally throughout asteroids.
How would we get in there? Maybe.
But then you look at
full
giant
of tardigrades is the other
is it
question.
full of tardigrades? Yeah. Well, and then you look at these massive asteroid belts and things that we have, and the abundance of ice that we found on asteroids, and then you can really start to maybe surmise it. Maybe this was a gas giant or two, or planetaries that have collided at some time. So it's either locked up or it gets dispersed amongst the debris field, but it's still there everywhere we look. Amazing.
Water everywhere. Not a drop to drink. So amazing. And physics, oh my goodness. It's only because of technology that we're able to even have looked at this point in time in our lives. So isn't that amazing? Justin, what'd you bring?
I like amazing things. Oh, that's not this one. Let's see. What do we got? Oh, this is a pretty amazing thing too. 15-year-old girl who needed a double lung transplant after years of fighting bacterial infection, and I believe she had cystic fibrosis as well. Got the double lung transplant. A few weeks later, she came down with a bacterial infection that spread throughout her body and liver. Infection was one that had, it was the same, I guess, that had persisted before the surgery, part of the reason she needed the lung transplant. But now it threatened to end her life despite the application of the organ transplant. The bacterial infection did not respond to antibiotics. These bugs didn't respond, says Graham Hatfull, Howard Hughes Medical Institute professor at the University of Pittsburgh. They are highly drug-resistant strains. So the doctors, was not Hatfull, doctors gave up on her. And then one of them was like, well, I know a guy who's been working on a thing. Let me talk to him first. And they got a hold of Hatfull, who's been working with viruses, specifically viruses that eat bacteria. He has a collection of something like 15,000 of these bacterial phages that have been collected by his students over the years. And yeah, so they basically created a genetically engineered bacteriophage cocktail. These viruses were introduced to the girl over 6 months. Nearly all of her infection had basically cleared up. The surgical wound began to close. These skin nodules that she had all but disappeared. Liver function improved. There was actually 2 patients that they were attempting to assist in this. And one of them didn't make it to the point where they could apply. And to sort of the scale and speed of this, they had within the lab found a bacterial phage that would work on that another teenager's particular strain that would overcome its resistance, that would kill it. They discovered it a month after it had passed. So this is sort of the time scale of this as a cutting edge science. This is why, if anybody's concerned that this is a sort of experimenting on children, these are verge of death, last arrow in the quiver opportunities. Yes. But yeah, the way they kind of went about it, they tested individual phages on the strains. They mixed thousands of these phages together, and they tested them, and they looked for ones that seemed to have this effect. They came up with 3 in the case of this girl, which they named Money, Zoje, and BPs. And one was really efficient. The other 2 kind of worked, but not as efficiently. And so they actually did a little bit of tweaking to them. Basically, what they did was they removed genes that let the phages reproduce within the bacterial cell, which meant that the phages reproduce and then have to escape the cell. They basically destroy the cell when they reproduce. They blow through. They did a little bit of testing before administering for safety, and then applied it. And yeah, After 6 weeks, a liver scan revealed the infection had essentially disappeared from the liver, at least. And 6 months later, she's doing much better. So Hafo has high hopes. Bacteria haven't shown any signs of developing resistance to these phages. His team has prepared a fourth one that they're going to add to the mix to try to get her all the way healthy in this case. But yeah, so right now it's a very individualized treatment because they have to find the right sort of cocktail of phages for each one, but they may be able to do this more broadly, of course, in the future. This is what you call cutting edge. I mean, this started, I guess this whole scenario started in 2017, and here we are in 2018 with a potential cure. Now, and this reminded me too of when we were in Portland. I brought the story about talking about how antibiotic resistance by I think it was 2050 is predicted then to have gotten to the point where bacterial infections kill more people than cancer, car crashes, and there was like a third thing combined every year. Yeah. It will become the number one killer. So the pressure is on, and in what better place to look for it than a virus, which is as we've also talked about on the show, where a lot of our immunity comes from. It's the toddlers have the highest viral load at age 2 because it's screening the body.
Yeah, but you're talking about a virus. This isn't just any virus. These are viruses that target bacteria. Yes. These aren't human targeting viruses. They target the bacteria. These are phages. They are specific to bacteria. Yeah. But I think it's amazing.
But that's what I'm talking about in the toddlers. That's what I'm talking about in the toddlers. Yeah, same thing. Those those are also bacteriophages that is when we come into this world, we have this high viral load that's there to eat bacteria. That's there to sort of filter for us to have a good start in life, microbiome wise, and that we shouldn't be now we're getting to the point where we can apply this later in life and bring back that initial start is brilliant.
Well, when you think about it, things like antibacterial resistance comes from this kind of one size fit all situation where you're just like, kill it all. Right. And there's also this universality of antibiotics like penicillin that were used very widely. Right. But it does seem like the more we get into modern medicine in this show, it does seem like more of a custom fit for each new patient, whether it be your microbiome or whatever kind of bacteria you're trying to get rid of or promote, or any of these other things, printing your own organs to order, right? So it's going to be a lot more, I think, successful to be looking at people as individuals in terms of kind of specialized.
It'll be good to be able to specialize, personalize on the infection itself. But I think like we've seen here, there's a lot of work that still needs to be done to like create the library of phages so that you have an ability to find out what are they infected with, and do we have the ability to treat it and not let somebody, pass because you can't nail down what they need for treatment. And then also, like Justin mentioned, they genetically modified these phages. They change them, and so you need time to do that as well. And so there are methodological concerns that still need to be tweaked out, but it's definitely this is coming. It's great.
And the thing that I'm not completely clear on, but I think part of this is that the reason it had to be individualized is because they don't have anything in the quiver. Right. So if they had somebody, if somebody came along with the same infection, they wouldn't have to start from scratch individualizing something for that individual person. They could apply what they had. They would have an arrow in the quiver for that combination of bacteria that they were infected with. So it's not that it currently has to be personalized just because they don't have the answer to any of those questions yet. They don't have those arrows, but they invented basically 2 in a couple of years in short work. So yeah, very promising field. I think I think we've talked about this before as a strong possibility for microbial defenses in the future.
Yeah, my hopes are on it. I mean, there's another story out this week that I ran across that salmonella. There's a Salmonella in some strains there's a jumping gene for antibiotic resistance to colistin, which is the antibiotic of last resort. And so salmonella is becoming resistant.
No.
Woo! Yay!
What a terrible way to go.
Yeah, but I mean, this is, we keep seeing this. The resistance is spreading. Oh, but you know what? It's time. Let's have some happy news. We have happy news.
Yeah, actually.
Okay,
Oh, it's about
great.
time.
It's time for Blair's Animal Corner. I think about now.
With Blair?
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair? And what was that I heard about except for giant pandas?
Those are later.
Okay.
Right now, I'm going to talk to you about wasps. Everyone's favorite insect. No, wait. This is a study from University of Michigan looking at how wasps do at logic puzzles. In math, in I think it was in middle school, I learned about the transitive property. If A is greater than B and B is greater than C, then A is greater than C. For a long time, we've assumed that is a human deductive reasoning that we use. Shock and horror, other animals use it. In recent years, we found that certain vertebrates like monkeys, birds, and fish have been able to use transitive inference. But this study in particular was looking at invertebrates, looking at bees and wasps. Another recent study found that honeybees could not figure out this transitive inference.
And honeybees are smart.
They are very smart. Yeah.
They do all sorts of navigational feats and they count, and honeybees are smart.
They can find their way to and from a location. They can communicate with each other where it is. But the explanation, no surprise, for a long time had to do with the size of their brain. So they have such little tiny nerve blob in their head. It's about 1 million neurons. They assumed could not exhibit this complex thinking. Paper wasps, turns out, have the same number of neurons, roughly about 1 million, as honeybees, but they fared differently on this test. So the test, this is going to get kind of complicated. I will try to explain it as best as I can, was to test this transitive inference. So they were given pairs of colors in a pen, in a 3 by 10 by 1 centimeter rectangle. One of the end of the rectangle had the quote-unquote correct stimulus, and the other one had the quote-unquote incorrect. The entire floor was electrified except for the 2 1/4 centimeter square closest to the correct stimulus, the safety zone. So they were trained over time with an A and B, a B and C, a C and D, and a D and E in these specific pairs of colors, which thank you to whoever wrote this, that they didn't actually use colors in their labels. They just used letters. Thanks, I appreciate that. That one was better than the other. So there would be one that did not, that had the safety zone. If they went to that color, that would be their safety space. So they were shown that one was better than the other. So for example, with A and B, A would be the safety zone, let's say. And then with B and C, B was the safety zone. C and D, C was the safety zone. D and E, D was the safety zone. So this is this, A is greater than B, or I guess D is greater than E, C is greater than D, B is greater than C, A is greater than B. So A is the highest, E is the lowest in this example I'm giving. So at the start of the training, they were in a clear plastic partition. They were placed in the center. It was turned on. They were removed. and the wasp was free to go around for 30 seconds. They entered the safe zone in every trial. So they never had to worry about, oh, they just never found the safe zone. After 30 seconds, they were removed. They were given a break. And then they went on to the next trial. They were given a 45-minute break before they tested learning accuracy after they learned all these. They did this for 5 days. And so they were trained which was better. They refreshed that training on day 5. And then they were given a 45-minute break. And then they got an untrained pair, B and D, or A and E. So they had to use inference to figure out which was the better one, even though they never found them together. So in this case, D was a safe zone, but B was not. But they still had to know that in this case, B was the safe zone because it was higher in hierarchy than D. I know that's really hard to understand.
I would have messed up the study. I'd have gotten zapped by going in the wrong.
Yeah. So in this case,
I'm not as smart as a wasp.
They had to make that leap of, even though this has been a safe zone before, it is not now because B's better than D, because B is better than C. I know it's a lot. It's hard for us to think about, but if there was an electrified floor, apparently we could figure it out.
Yeah, I think there might be a learning curve with that. Yeah,
electrical shock, maybe.
I would have studied harder in school. I really would have.
Yeah. So
would have done better on those tests of transitive inference. Yeah.
They picked the correct corner more often than by random chance. And this shows that they have some idea of this inference. So they have this way better understanding of how things are related. Now, the reasoning for why this might be the case with wasps and not bees is because they have different social structures. So a honeybee colony has a single queen, and they have multiple equally ranked female workers under her. In contrast,
in terms of hierarchy,
yeah,
that's not much. It's somebody at the top. The rest of us are all equals here.
Exactly. So in contrast, paper wasps have several reproductive females. We call them foundresses. The foundresses compete with their rivals and make a linear dominance hierarchy, just like this. So in theory, if you found foundress B and foundress D, you would know that D is higher than foundress E, and you'd know that foundress A is higher than foundress B. But if you wandered into one area where there was B and D, you would still have to pay more attention to foundress B.
Mm-hmm.
So you'd have to be able to figure out this transitive property through hierarchy,
or at least the females would.
Yes.
So is that, and so the question then is across, I guess this would be across all the wasps, or is this somehow a gender determined?
That is a great question.
Is it better or worse, or is there a range of abilities based on?
Are you indicating
role in the social structure
that contrary to popular opinion, for many, many, many millennia, females might be better at math than males? Anyway, paper wasps recognize.
Why is that contrary to many millennia?
Oh, because girls are bad at math. That whole joke. Anyway,
we're not.
I have no idea what you're
talking
about.
Turns out we're not.
Who said you were?
Not only. Oh, just decades, millennia of men. Don't worry about it.
Don't listen to men. They don't.
Yeah, they're bad at math.
Yeah,
so, anyway, these paper wasps need to be able to tell each other apart. They also have been shown to recognize individuals by their facial markings, so that they're kind of set up to recognize this difference between females in that way as well. But this is mainly just setting up that even invertebrates, animals way far away from us on the evolutionary tree, need to be able to do this complex, what we would consider logic thinking, these logic puzzles that we for so long assumed was only something that humans could do.
Animal Kingdom strikes us down once again.
And if you had trouble following that, I'm sure you're not the only one. I am a visual learner. It took me reading this study a couple times to understand what was going on. Go to TWIS.org and find the show notes, and you can read a little bit better. You can send me an email or tweet at me. I'll try to explain it better. Anyway, moving on to fly sex. Fly sex. So fly sex is interesting for a bunch of reasons. They do have internal fertilization. And we've known before that, and I think we've talked about it on the show before, that male fly ejaculate makes females lose interest in other partners. And I think we have had studies on the show where we've talked about is it hormones? Is it neuronal path? What is going on? But this new study from Howard Hughes Medical Institute was looking at another element at play in a female fruit fly's disinterest in other males after copulation. And that has to do with the mechanics of how they actually copulate. So when a female fly has uninterrupted sex, a pair of neurons exclusive to females carry a stop mating message from sensory neurons in the abdomen up to the brain. This has been dubbed the copulation effect, and might be especially important in the wild when matings can be interrupted partway through. The head researchers on this were actually hunting for neurons involved in reward circuitry when they stumbled across this finding, which I think is very funny. They noticed that certain cells in female flies sent strong reward messages to the brain when they were stimulated, but those cells didn't even exist in males. So they said, okay, this is something sex specific. Let's look at it. In one experiment, they put female fruit flies with males that couldn't ejaculate. So there was no sperm signaling in there. After mating, the females lost interest in other males, even though they got no sperm from the males. When they blocked this particular neuron's activity that they found, the females kept trying to mate. So these cells control a way that female flies determine when they've successfully had sex, independent of the sperm effect. Why would this happen? This seems disadvantageous if you're a female. Well, the sperm effect, which is the one that's kind of the chemical response to the ejaculation, takes a while to set in. So this kind of bridges the gap in between copulation and the hormones or whatever it is from the sperm signaling them to stop having sex. This is actually a very short-lived response from the neurons. And so it sounds like most likely that takes over for the short term until the sperm effect can kick in.
Is it? But are we still sure the sperm effect is at play then? Though I mean, if it looks like there's a whole other mechanism that's doing the job.
So it's kind of an overlap. So the copulatory effect is the short-lived, immediate, and the sperm effect takes on later and is more long-term.
And okay, good for those who can't see.
But because they have
Blair has done a giant hand diagram of overlapping things.
Yeah, overlapping things. It's like, go ahead.
I was just going to say, I mean, part of the experiment was controlling whether females got sperm or didn't. And so so when you see that there are some female who did not get sperm, but like the copulatory effect stopped them, and then that wore off, and then they maybe wanted to mate more quickly than those without sperm. And you've got these two effects that you can see.
There you go. It's like, to try to give it a metaphor, it's like I had a migraine on Monday and I took some pain medication, but that takes a while. It takes about 20 minutes to start working. So I got a nice huge ice pack and I put it on my head. That was a short-term solution because it just kind of numbed what was happening superficially, and it happened long enough so that I didn't barf everywhere until the pain medication kicked in about 20 minutes later.
Fun story.
Yeah. Short term versus long term effects. There you go.
Short term versus long term.
Uh-huh.
And for those of you in the listening audience, when she said short term and long term, Blair did a short span between two fingers, and then followed by a longer span between her two fingers.
What would
Same
I
two
do without you?
Oh, what would we all do without each other? Well, you're going to get a chance to find out for about a minute or two because it's time for us to take a break. We're going to take a quick break so we can have a few messages. And then when we return, we have more stories for you. We have a What Has Science Done for You Lately, collapsars, and much more. So stay tuned for more This Week in Science.
Can't explain things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need.
Thank you for listening to This Week in Science. We come to you every week, and it is such a gift that you join us, that you bring us into your lives on a weekly basis. But you want to know how you can help continue to make that an ongoing event week after week. We do have to support this show and pay for the many ways that we make the show happen and fun things that we try to do. So if you are able, we would love your support to keep the show going. If you're interested in how you can support the program, head over to TWIS.org. TWIS.org is where all things TWIS are found, links out to other places and resources, show notes, all the fun stuff for each of our show episodes. If you have a question about stories, that's where you find it. But also at TWIS, you can find things like our Zazzle store link. At the Zazzle store link, click it. It'll take you to our Zazzle store where you can find all sorts of TWIS items, t-shirts, and polo shirts, tote bags, pillows, phone covers, mouse pads, wonderful items with the TWIS logo or art from various Blair's Animal Corner calendars. Many items in the store, and there is definitely something for anyone's needs that you can find there. Do you need a baby onesie? We have those TWIS onesies. They're so cute. Or wrapping paper? There is TWIS wrapping paper. It is true. Need a coffee mug? Oh, that's where you find it. And a portion of these proceeds do go to support TWIS. And so we thank you for any purchases that you make there. Back at twis.org, you can also click on the donate button. There's a little yellow donate button on the side of the page. Click on that donate button, and it will take you to a PayPal interface where you can choose an amount to donate. It's a one-time donation. Just click and pay through PayPal to support us at the amount that you are able. If you would like to support us in an ongoing fashion, click on the Patreon link. Patreon is a crowdfunding community for creators. And over there, you can click the red button. Lots of clicking going on here. Click, click, click. Become a patron. Click that red button and choose your level of support. You will be charged once per month for whichever level you choose. $10 a month and more. We will thank you by name at the end of the show. Wouldn't that be fun? There are all sorts of other fun things you can get through the Patreon community also. Finally, back at twis.org, there is a big orange subscribe button. It's just for the big three, Google, iTunes, and YouTube, but it's an easy way for you to subscribe or for you to get someone else to do the same. Send someone to twis.org to subscribe. You can also find us, though, at fun places like Pandora, Spotify, Spreaker, radio.com, TuneIn, all the big places where you can find podcasts. We are there. We have a Facebook page, facebook.com/thisweekinscience. And if you're in Santa Fe or thinking of heading to Santa Fe, we will be there June 14th or June 15th and 16th for the Interplanetary Festival. We are going to be podcasting on June 15th. Can't wait to do a podcast at the Interplanetary Festival in Santa Fe, sponsored by the Santa Fe Institute. Thank you to all of you for your support. And honestly, we really could not do this without you. Thank you.
more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go.
And we're back with more This Week in Science.
Yeah, we are. You know it. We have more science for you. But first, what has science done for me lately? This letter-
Beautiful.
it's always beautiful. Such harmonizing. That's right. All right. This letter this week comes in from Minion Dale Moore, and Dale wrote in to say, 6 months ago, I had to take a Toyota Prius to the dealership to get a headlight assembly replacement. I had to leave the car there as the maintenance staff explained that it was about a 5-hour job. Fast forward 6 months. Yesterday, a close family friend went into surgery. He has been battling brain tumors for a while now, undergoing traditional treatments as well as gamma knife. Last week, he became numb on his left side and started exhibiting signs of a possible stroke. After going into the hospital, he was told that a right hemisphere brain tumor had increased in size so suddenly that it was impairing his motor functions and a surgery would have to be performed as soon as possible. Possible side effects were partial to total paralysis on the left side. He went into surgery yesterday afternoon, placed under general anesthesia, had his skull removed in the area, tumor removed, and in recovery in 4.5 hours. As of last night, he was still unable to speak clearly, but had movement on his left side and was able to tell with sign language that he was okay and signed "I love you" to his wife. The man was in and out of a successful brain surgery faster than the local car dealership could fix my headlight. What a time
Wow.
to be alive.
Mm.
Yeah. Unfortunately, I did get news from Dale today when I was informing him that I would be reading his letter that this friend of his has passed away due to complications from the cancer. But when I asked him if he wanted me to still read this letter, he said yes. And so the thought, he said, is still there that we really are in an amazing time to be alive. And so, Dale, thank you so much for being a member of our community. Thank you for sharing this story with us. And we wish the best to your friend's family and to you. Thank you. If anyone is wanting to write us a letter, a song, a sonnet, you can do that. We need you to help fill up this moment in the show. What has science done for you lately? Please help fill it up by sending me whatever story, sonnet, song, whatever, to my email, Kirsten@thisweekinscience.com, or send it to Facebook, facebook.com/thisweekinscience. We do want to keep filling it up. And Blair, there's a song coming. Not now, but I have an email. It's scheduled. I've got it on the list.
Oh my God, I'm so excited.
There's a song. I don't know that we'll be able to play it for copyright reasons because of the music used, but there's a song on the way. There is. Seriously, what a time to be alive. Blair, not you. Wait, who's got a story next? What's going on in the
I could
world?
go if you wanted.
I got one.
Where am I today?
I got one.
Justin, you have a story?
Yeah, this is about the Cambrian explosion, which a lot of people don't remember this. It was about 540 million years ago.
Oh yeah.
And it's sort of the key time frame for the evolution expansion of complex life forms throughout the fossil record. That question has been around for a while, though. Why did that happen? Why will so much life get so complex and so?
It's usually something has space, and then a bunch of evolution is like, how about this? How about this? How about this? How about this?
So has been the sort of predominant common theory, is that it was a sort of tipping point for evolutionary niches and expansion into those niches that was taking place. And that's of course what happened. But there's more to the story, it seems. There's a multidisciplinary study published in Nature Geoscience by a joint China, UK, Russia research team. And they're giving an argument that oxygen content of the atmosphere and ocean was the principal controlling factor for the Cambrian explosion. So there are in this record, there are patterns of radiations of complex life, and then sort of a mass extinctions that would also follow, sort of a back and forth. So it wasn't just one directional explosion. It was wave upon wave of forward and back. And it turns out the pattern of going forward and back coincides with fluctuations in the carbon isotopic composition of seawater, which is correlated with the production of oxygen in the atmosphere and in the oceans. So it now has added another layer to our understanding, perhaps, or another layer of questioning to examine exactly what is important for life. And I think this is this story is interesting and exciting in a lot of ways, but it also is very crucial right now as we talked about the decline of corals. As we learned on this show, and much to my surprise, corals produce half of the oxygen on the planet. And so the decline of corals means decline in oxygen. And we now know that alone is tied to mass extinctions events on the planet in our history. So lessons learned are lessons learned if you act upon them. If not, they are mistakes repeated. Yeah. How that works. It's pretty simple. Point. It's straightforward. Yeah.
Good point.
That kind of, it does remind me of the winged, the bat-like winged dinosaur that looks a little bit like a dragon in the pictures that I've seen in the illustrations of it. But it predated the dinosaurs that turned into birds, the winged dinosaurs that became birds. And so it was kind of like this period of time where evolution was trying on wings and going, how does this work? How does that work? Where does that? And so it's one of the styles. It's one of the dinosaurs that didn't make it.
But what's interesting about it too is it makes me think of, okay, maybe not now, but let's file this away in the genetic data bank of a thing we could do at some point again, because
we have it in there.
Yeah, because as a lot of what we learn about evolution we keeps coming up to, it's not necessarily a new thing in the history of a life form. If you follow it back, it's a reemergence of a thing, or a reapplication of a thing is also something that takes place in evolution, which is very like when they but
at some point these things are new.
But we can dial back. We can dial back embryonic chickens to having teeth, right? So the information is there, and there could be a day in the future where humans die off, nobody's feeding the chickens, and they start going after different prey, and they get teeth again. It's in there. They have the genes to produce teeth.
Right. When chickens bite back.
Right.
They already do that, and it already hurts.
But yeah, genetic evolution and mutation and all this also files away for future reference, and so we do see these reemergences as well as convergent evolutions and new traits that are picked up along the way.
New and different, yes. Well, there are lots of different kinds of stars out there. I love stars. There are many different stars. Have you heard of collapsars before?
This is not a thing I've heard of before. That feels like what I do on a dance floor, because I refuse to get off the dance floor, and then at some point I just become a collapsar.
I'm a collapsar. That's what I am. No, collapsars are these massive spinning stars. They're spinning around, and giant, massive, and then they collapse into a black hole. And so when they collapse, they go supernova. And it's that supernova that releases a massive gamma ray burst and also explodes out those outer layers of dust and gas off of the star out into a nebula and out into the surrounding space. And this is the process by which this implosion, right? The supernova explosions, by which heavier elements are thought to have seeded the universe. And researchers have been trying to figure out where exactly all the gold, platinum, and uranium in our universe came from. And we always make jokes, right? Oh, the big stars. Well, maybe that's where the gold came from. That's where the stuff that came from. But till now, we really only have had evidence of a merger of neutron stars possibly creating gold that would be thrown out into the surrounding universe. Lighter elements have been discovered all over the place, but there's a chain of reactions. It's called the R process. That really only happens when you have densely packed material, neutrons, right? So this R process happens, and in the R, it's not a pirate process.
But if they're going after gold, I don't know. I feel that kind of fits.
It's the R process. Atomic nuclei absorb neutrons really quickly, and they undergo radioactive decay. And in this R process, new elements that are heavier get created. And so they suspected when neutron stars collide, this happens, and then astronomers saw neutron stars collide. We recently saw this because of LIGO, and we were looking at it, and oh, hey, look at that. And they have evidence for these elements, gold, silver, and platinum, as a result of seeing this neutron star merger. However, neutron stars can't tell the whole story. And so researchers were like, well, what else could it be? Well, it's got to be collapsing stars, but we haven't seen this before. And so there's a new study that was just published in Nature on May 6th. Researchers have been looking at these R processes in stars and expanding on a study from back in 2016, discovering a dwarf galaxy called Reticulum 2 that they thought resulted with these elements in the galaxy resulted from a neutron star merger. But now, as the result of this study, they think it's a collapsar, that it was a massive spinning star that supernovaed and spit its elements everywhere. They think, as a result of their analyses of these massive stars, that the collapsars might actually be the things that are responsible for 80% of these elements that come from the R process, the gold, the silver, the platinum, the uranium, in the universe. And that neutron stars might only make up about 20%. This is the new idea.
But still, I would still think the neutron stars would be better at spitting it out.
Right. But the neutron stars, they could possibly be better at spitting out the elements. The massive stars are just bigger, though.
Yeah.
It's just because they're so big, they contain so much more stuff that the abundance of material is so much greater that's why they think that these collapsars are what are responsible.
It's more localized. So, sorry, I'm doing a stupid statistical analysis of across the universe where the gold is. Right, yeah.
Right.
That's fascinating.
Yeah, so with more studies, they'll figure it out. How much is collapsars? How much is neutron stars? We still don't know. Is there another process that we still haven't figured out yet? Because there might be that as well.
Yeah, but both of those, the high pressure thingy that creates these, makes sense.
Yep, high pressure, high temperature, massive event taking place. Yes. Tell me another story.
Oh, is it me again? Okay.
Yes, that's what happens.
Oh, no,
you tell a story, I tell a story, you tell a story, I tell a story, and then Blair tells a story.
This is at last a human origin story that I didn't like. This is, but I'm just going to read it as it is. Statistical analysis of fossil data shows that it is unlikely that Australopithecus sediba, that's the hominin that was found in the cave, the deep under, where they had to hire really small archaeologists and anthropologists to go spelunking for in South Africa. The one that looked like they were doing a burial, like they might have been dragging the dead there, because there was a lot of all across all ages, like you would see in a cemetery, and there weren't kills, there weren't animal bones. It didn't look like they were in there eating and living. The only evidence was these populations being dead. This research is by paleontologists from the University of Chicago. They're publishing in Science Advances, concluding that Australopithecus afarensis, aka the famous Lucy skeleton, is the most likely ancestor to the genus Homo, and not Australopithecus sediba. So, yeah, these were it's the oldest known Homo fossil. Jawbone is yet unnamed species found in Ethiopia. Is 2.8 million years old. They got the jawbone. They don't really know the rest of it, but it's 2.8 million years ago. They're saying this is a Homo from this, and it predates Sediba by about 800,000 years. That's the current Sediba find. That's the oldest jawbone find. But again, it's an unnamed species. So they're saying, yeah, it's very likely, because the average run, I suppose, for a hominin is about 1 million years. So they're saying it to be at 800,000 years already. They missed the mark by 2 million years, or no, by 800,000 years. They're probably not the predecessor of it. So despite this timeline, researchers who discovered Sediba have claimed that it is an ancestral species to Homo. While it is possible that Sediba could have post-dated earliest Homo by 800,000 years, new analysis indicates that they probably finding this chronological pattern is highly unlikely. So I guess there's a couple of things here that sort of bothered me in this. The statistical analysis is based on, one, a kind of unidentified Homo jawbone, which you could have possibly done with something that was very disparate. It sort of begs the idea, the old way of thinking, because they're not using genetics or anything like this. They're using morphology, some statistics, and just the data points available to come to a conclusion. What they sort of lack in this, though, is an understanding of that whole braided stream concept. The idea that this could have been a completely different offshoot, right? Like this could be another hominin that is also not related to Homo, but is morphologically similar. That there could have been a bigger experiment, even going back further between the different hominins that were around, so that you could have something that could have morphological similarities in different places, but still not be the Homo ancestor. So based on everything that we've learned in just the last 5-10 years, it seems like that's a giant conclusion to make with very limited data. But still, interesting.
I feel like a lot of, this archaeological, paleontological data is, you small sample sizes, and it's just these years and years of work to just get a few more bones and hopefully get enough data to be able to make these broad statements about our evolution, right? And but it's neat the methods they're using these days to be able to actually do it. How long have humans been around now?
Oh, it goes back and forth, but I guess the rough is about 200,000.
Okay, so we've still got, unless we kill ourselves with climate change, we've still got about 800,000 years left.
Yeah. Oh, but you know what? I got to take that back. It's got to be older than that, because there was an interbreeding event with Neanderthals,
right,
which massively predated what we thought, which would put it more in the 300,000 to 400,000 years.
Okay, so we're still we're less than halfway. It's great. We've got time. It's good.
Well, there's other factors. I mean, if we were doing nothing but running around being hominins, yeah, okay, got a long run ahead of us.
Who's
We're
to say
bringing
we're
about
not, Justin?
Cambrian era type extinctions by reducing the oxygen with the knocking down of. You know what? People smoke them if you got them. It's fine.
Hominids got a hum, you know?
Hominids got a hum. There, yep, there you go. Okay.
Okay.
Words to live by from Blair. All right, moving on to my last story. So I was thinking about bringing a story about lung regeneration because I was like, hey, that's cool. We can potentially regenerate lungs for smokers, but that's not really what it is. It just is a method of regenerating lungs that allows them to last a few hours longer. So it'll help with being able to get lungs to patients who need transplants, which that's kind of cool. But there was this other news that I thought was really exciting about some researchers from King's College London have been working. This is also researchers from the British Heart Foundation, the head of the School of Cardiovascular Medicine and Sciences at this King's College London. It's an international collaboration, really, of also involving researchers in Italy who have been working to help repair the heart that has been damaged from heart attack, myocardial infarction. Now, in a heart attack, the muscle is damaged. It doesn't receive oxygen. The muscle cells die. Those cells then form a scar tissue, and that scar tissue impacts the ability of electrical signals to travel through the heart and also the ability of the heart to pump. Cardiac arrest is one of the leading causes of death today, if not the leading cause of death today. And if we could repair the heart after heart attack for those people who survive, it could be amazing. It could change lives and make lives longer and more fulfilling. So we've got these different methods, researchers trying to do heart patches and stem cell patches and different things. Well, these researchers who published this week in Nature, again, have published their study using pig hearts, which are very similar and about the same size as human hearts, in which they used a new generic compound, microRNA, it's 199A. And they treated infarcted pig hearts with this microRNA 199A and found that it stimulated cardiac repair. The cells in the heart started repairing the damage. New muscle cells were formed. There were proliferations of cells. And 1 month after myocardial infarction and delivery of the RNA, and they used an adeno-associated viral vector. So again, this is a viral vector that would probably have to be looked into for human use. That's not an issue when you're dealing with pigs, but it allows this microRNA to get into the cells to actually have its effect. These treated animals showed increased muscle mass and reduced scar size, improvements in global and regional contractility. There was healing taking place in the hearts, which is very exciting. However, bad news is there was no control over the proliferation of cells, and so the microRNA, the microRNA just stayed there and kept turning the cells on, and so more and more cells were being born, and they got into the wrong places, and eventually the pigs died of arrhythmias because their hearts stopped working well. But the study does show that there is promise in this kind of method if we can learn how to control it. It's like we put it in and the cells, they start dividing and things start fixing, and then it's gone too far. It's not stopping. Yeah. that was the story. Yeah, so it's potentially a very exciting therapy in the future, but the fact that it was successful in a certain extent of remediation of the damage is very exciting, and we will see where it goes in the future. What will we see? Oh yeah, and I got a couple of quick news stories. We ready for some quick news?
Oh, yeah.
Mhm.
Yeah. So, first quick news story. There is a new blood test for Alzheimer's disease. And when it looks for one of the proteins, the misfolded proteins in the blood. So it's not super invasive, just a blood test. And it was able to, within about 70% accuracy, be able to determine whether or not somebody would have Alzheimer's disease as early as 8 years before any sign of symptoms.
Wow.
And the researchers fiddled with their tests a little bit more and found when it was paired with a test for a different protein, the tau protein, that was taken from cerebrospinal fluid, that those results went up to 97% accuracy. They are hoping that they will be able to get this blood and cerebrospinal fluid combo test out into the market soon, because the potential to be able to find out whether people are starting to get the accumulation of proteins that will lead to the symptoms of deteriorating mental function that are associated with Alzheimer's disease before those symptoms, years before those symptoms ever start, means that there may be hope for treatment.
Yeah. See, that's the key, because
Happen early.
we weren't getting as far as we have been with Alzheimer's treatments, potential treatments, I would say, I don't know if you'd want to know. It would be tough to kind of have this ticking clock knowing that it's going to start happening. But since we have avenues to potential treatment, this is huge.
It's huge. And it's also been discussed that some of the antibody treatments that failed in trial, researchers think that it's possibly because they were testing on people who were such late stage Alzheimer's. It was people who already had massive amounts of damage, and that perhaps if it were with patients who are accumulating proteins but still years away from actual symptoms showing up, that maybe the damage might not be at a point yet where it's too far gone to treat. And so maybe it might be worth revisiting some of these possible antibody treatments.
Yeah, that's a great point.
Yeah, there's a story we didn't bring that has a link between doing diagnosis and then treatment, but also of some spectrum of schizophrenia being able to diagnose by the blood. So we're finding it sort of seems like a blood neural connection in there that is, yeah. We'll have to maybe look into bringing that story next week.
Yeah, that'd be
it
cool.
Seems like this is an interesting blood-brain connection.
There is. As much as we think our brain is separated, with that blood-brain barrier, there's still some crosstalk, and there are things that we can find.
And apparently the blood carries the evidence of what it's seen. It's taking pictures along the way.
More like a blood-brain sieve, am I right?
I was going to say blood-brain tourist, but sure.
Yeah.
And finally, there is a study out this week in Nature Human Behavior about Pokemon brain.
What?
That's right. Turns out that people who played Pokemon as kids had developed a very unique region of their brain that is specialized for recognizing Pokemon characters.
Oh, my goodness.
So you've heard of the grandmother cell, or these cells in our brain that respond to individuals, faces of individuals that we know, or that it allows us to recognize important figures very easily in our daily lives. And those individuals who have grown up with Pokemon characters were very important. And so there's the region of the brain with neurons dedicated to Pokemon characters. And the region of the brain and the neurons seem to be correlated with the size of the device that they used, which is associated with the size of the characters on the screen. Yeah.
So I
That's
just
a-
wonder if this is similar to, do I have an area of my brain that is focused solely on recognizing types of animals by the way that they look? Or does
Possibly.
someone who loves trains have an area of their brain that is focused on recognizing different types of trains? I feel like it has to be the same stuff.
Yeah. Is there what you focus on, what's important to you? Your brain is specializing for that, especially during that period of youth when your brain is developing and determining what is important.
Yeah, just like a categorizing enthusiast.
Mm-hmm.
Like a category building brain space. I don't know.
Yeah. So anyway,
Speaking of things about animals in my brain, pandas. That was
pandas.
flawless transition. Pandas, they their guts might not be so stupid. We know they are specialized herbivores. They eat bamboo, but they have come from a crop of carnivores from other bears and other carnivora family members. And so we've talked before about how their gut is directly related to eating meat. And so we kind of thought that was something that showed that they were just wrong somehow. They have a digestive tract, digestive enzymes, and gut microbes that are for carnivores, not herbivores. Well, looking not just at the food that's going in, but the nutrients in the foods that are going in, they have found that despite their plant-based diet, the protein and carbohydrate content of that diet looks much more like hypercarnivores, animals that obtain more than 70% of their diet from other animals than that of herbivores. So about 50% of the energy they get is from protein, and I did look it up, and bamboo is a very high protein plant. It contains between 1.5 to 4 grams per 100 grams of bamboo shoots in protein. So that's pretty high. And so I guess it ends up that their nutrient intake is actually quite similar to if they were just eating meat.
Wait, so could we all replace the meat in our diets with bamboo?
No, we could not digest bamboo, but
well,
good try.
Well, unless we had the proper microbiome.
There you go.
We could do a microbiome
Panda poo
transplant.
pills. Panda poo pills.
Panda poo pills. Everybody
Yeah,
get them.
yeah.
Dr. Justin's not a real doctor. Panda poo pill. Emporium.
Making bamboo not just for your floors anymore.
Yes.
Yeah. So the implication from this is that the evolutionary track between a bear that eats meat and a panda is that evolutionary distance is actually quite minimal, and the transition is in fact more superficial than we thought. And that also explains why they have this weird mixture of herbivore and carnivore traits. Might not be nature's mistake. It might be just fine.
Oh, are you
heard it here, folks.
Are you rethinking
Blair?
your love for the panda?
No, they're
Blair,
still bad at reproducing and finding their own home. So
Okay.
I've
All
had
right.
it still.
The two things that are the highest on Blair's list of qualities in a good animal.
You
Finding
must
a
be able
home.
to- yeah.
And must reproduce.
That's the name of the game. That's why we're all
Prolific.
here.
They're
Propagate
still here. A
DNA.
lot of things aren't here. Pandas are here.
Because of humans. Humans did it. Okay,
Well,
moving
because
on.
we didn't kill them off. Is
We're
that-
Moving
saving them.
on.
We're actively
Tell us-
saving them. Moving it.
Tell us
Oh,
about
oh.
your lost stash.
Oh, so yeah, this is what is this? This is University of California, Berkeley archaeologist Melanie Miller. The chemical analysis of a 1,000-year-old pouch made from 3 fox snouts sewn together. That was found high in the Andes Mountains, I believe. I'll get to this. They included dimethyltryptamine, DMT, and harmine. I don't know what this is. Active key components of ayahuasca. Mind-blowing.
Ayahuasca.
Ayahuasca, mind-blowing. Yeah, so obviously I've been out of the hipster scene for a while. Ayahuasca, mind-blowing brew commonly associated with the Amazonian jungle. Quotey voice of Miller. This is the first evidence of ancient South Americans potentially combining different medicinal plants to produce a powerful substance like ayahuasca. Let's see. So they did analysis of scraping from the fox snout patch, plant sample found in what they're calling the ritual bundle. Why are all ancient people constantly doing rituals? Is it possible that this was just like a day tripper? Like, why?
To them, it was just Tuesday. Yeah.
These days we call it a festival fanny pack. No.
But this could have just been this could be normal life.
Could be.
There wasn't, media distraction, music that you could just download and listen. It's you had to be entertained back in the old days. This might have just been it. Discovery adds to a growing body of evidence of, again, ritualistic psychotropic plant use going back millennia. And then this is what I sort of find is sort of funny about this too. So let's see where it is here. They were found at 13,000 foot elevations in the region of southwestern Bolivia. This is picture wandering llamas and alpacas. That's about where this would be. The kit itself goes back 550 to 950 AD, pre-Incan Tiwanaku civilization, which used to dominate the southern Andean highlands. In addition to the medicinals, they found a carved wooden snuffing tablet, a snuffing tube that had human hair braids attached to it for snorting intoxicants. They found some llama bone spatulas, colorful woven textile strip, and dried plant materials. A very basically a junky stash kit of sorts. Sounds like. But the where was this? What was sort of interesting is that they have thought of the site where they found these to be a burial site. They thought it was a burial site, which would fit with the ritualistic nature of having this is some sort of communing with ancestors. So they did an excavation. Guess what? No human remains. This is just somebody up at the altitude having their version of fun. Again, but then
they're having a wander, right?
Yeah, it says plants found in the bundle do not grow at those altitudes, so it's not like they went there to collect them. But then automatically, here we go, suggesting the bundle's owners may have been a traveling shaman or some other expert in rituals of psychotropic plant use. The thing is, people of yesterday were not different than people of today. People of yesterday were the same people. They had different experiences, yes. The cultures were different, the technology was different, but the people are people. Not everyone going around with psychedelics in their back pocket, in their little pouch, is doing a ritual or is a shaman magic man that's wooing people. This person maybe did know how to provide proper doses so that people would not be hurt or harmed. This is typically today known as a drug dealer, but it does not mean that the people around them were wowed by the magic shaman's pouch of wonders. They were probably knew what they were expecting at least the second time around. So, anyways, really interesting find. But please, not all ancient people are doing rituals. Unless you want to start defining everything that we do in modern society as rituals. Oh, we're here for the ritualistic throwing around of a pigskin, which is a ritual we currently have. Nobody talks like that. Stop. Anyway.
We love the rant that comes along with it. Oh, everyone, I believe we have come to the end of another show. Have we done it?
Yep.
We did it.
We did it. Yay, everyone. Thank you. Thank you for a great show. To you out there, thank you for joining us tonight. Thank you to all of you in the chat room for chatting with us. Thank you. I have some shout outs to give to individuals, to Identity for helping us record the show, to Fada for helping keep our chat rooms nice, and for social media, and also for show notes, and Gord Gord McLeod, thank you for keeping our chat room wonderfully kind as well. I do appreciate that. Thank you to our Patreon sponsors, which of course I got halfway through putting them all up on the page in front of me, and then I got into Justin's story.
What?
I was listening to your.
Oh.
I was
It's your
listening to your
fault for
story. I got
being
distracted.
interesting.
Interesting.
I was
Oh my
listening to.
goodness.
It was pandas and then ayahuasca, and I'm like, what's going on here? And then I yes, okay. Thank you to Paul Disney, Richard Onemus, Ed Dyer, Andy Gross, Stu Pollock, Philip Shane, Ken Hayes, Harrison Brothers, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Stony Steele, Craig Landon, Mark Mazzaros, Jack, Matthew Litwin, Jason Roberts, Bill K, Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Corinne Benton, Adam LaJoy, Sarah Chavis, Rodney, Tiffany Boyd, John Bertram, Mountain Sloth, Seth O'Gradney, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Sue Doster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Artyom, Rick Ramus, Paul, John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slizewski, Jim Drapeau, Greg Riley, Sean Lamb, Ben Rothig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie, Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and E.O. Thank you for all your support on Patreon. For those of you out there who are interested, you can find information about Patreon, a link to it at TWIS.org. Remember, you can also help TWIS out by subscribing. Tell your friends about TWIS. On next week's show, we will be expecting a guest. We are going to be speaking with a researcher in New Zealand, Chritina Hulbe, who looks at Antarctic ice. And so we'll be talking about ice in Antarctica and what's going on with those ice sheets down there and what her research is telling us. Very excited about that conversation. And once again, we will be broadcasting live online at 8 p.m. Pacific time, twis.org/live, or on YouTube. YouTube? No, YouTube. YouTube. Both places you can watch live and join the chat room. Don't worry if you can't make it, though. You can find our past episodes at twis.org or on that YouTube channel.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory. Or if you have a mobile device, you can simply search for This Week in Science, and we should pop right up.
For more information on anything you've heard here today, like if you want to see that diagram about the wasps one more time, show notes will be available on our website. That website is, of course, www.twis.org. While you're there, you can also make comments and start conversations with the hosts or other listeners.
Yeah. Or you can skip the other listeners and contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line. Otherwise, your email is likely to be spam filtered into oblivion. And you can also hit us up on the Twitter where we are to science at Dr. Kiki at Jacksonfly and at Blair's Menagerie. We love your feedback. If there's a topic you'd like us to cover or address, a suggestion for an interview, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. Science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause Cause it's This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. Science. This Week in Science. This Week in Science. Science. This Week in 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 Science.
And that brings us to the after show. The after show is after the show. It's the time that we're after the show, faux show. It's not the show, it's the after show. It's not the pre-show, it's the after show. After comes after, pre's before. It's not the before, it's the after. We're done. What not done? It's, I don't know. What are we doing now? Hey.
What even are we doing?
What even are we doing, man? So there was a story that Phage over on YouTube, in the chat room over on YouTube. Oh, he's going away. He said goodnight all. He was wanting to talk about, and I don't know if he had posted it to our Facebook page, but there's another newly named Tyrannosaur. It's Tyrannosauroid. It's a relative of the T-Rex, but it's bitty. It's a little tiny T-Rex.
Tiny T-Rex.
Yeah, about 3 feet tall at the hip, 9 feet in length. So it was short and long, but still T-Rexy. So cool. To find a picture of it. I wonder if I just looked up bitty T-Rex.
Witty bitty.
T-Rex relative. There you go. Look at my Googling skills. Let's see. There it is. T-Rex cousin. Live Science says, newfound mini T-Rex was a tiny terror at just 3 feet tall. Someone else says, these T-Rex relatives are an evolutionary stepping stone. 3 feet tall.
Do you have a picture?
I'm
You have a
looking for it,
drawing?
Yeah, I just don't want to do Live Science, so I want to find a different one. Live Science has too many ads on it. I need to find a different source, and I can find pictures. New Atlas, maybe. I will find it. Hold on. Hold on. Yeah, I'll just use New Atlas maybe.
All right.
Everything's got too many ads here. Oh, we got lots of pictures. Here we go. I'm sharing the pictures. Let's look at this cousin.
What we look at?
We're looking at bitty bones.
Tiny
The-
T-Rex.
Look at the jaw is in his hand. Look, there's a T-Rex jaw. This is a real T-Rex, and this is this 3 foot tall T-Rex that was found. Look at that. It's so cute.
There's a little baby T-Rex.
And then here comes another one. Look at that. There's a researcher with the little bone of.
Look at that
This-
shirt. He matched the. He matched the.
He's matching the
feet
back.
it's
Look
laid
at him.
out.
That's right. It's
Yeah.
probably school colors.
That would make some sense.
Look at this nice. Look at those bones.
Wow, that's
Look
a
at how-
good. That's a.
It's a
Yeah.
great skeleton.
Yeah.
Yeah, you've got little claws from the forelimbs, and you've got the pelvic bones back there. We've got legs. We've got a lot of vertebrae. The
That's a
jawbone.
lot.
Yeah, that's pretty cool.
Nice.
And so these fossilized bones, I'm trying to find the image of what they think it looked like. Oh, so he was 16 when he found the fossils in 1998. Okay, that's him then when he was 16, and then that was him up at the top in his color coordinated shirt. So he's been working on this fossil for a long time.
Wow. That is very cool.
Yeah, it is.
Yeah. Oh wait. Yeah. Yeah. It's right. There it is. Okay. So they
Look
thought
at
it
it,
was
it's all fuzzy.
fuzzy and feathery,
Oh my
and
god.
it was named
That's amazing.
Suscatyrannus hazelae.
Oh,
He
it's-
does
Look
look
at
kind
her.
of like a bird. He looks like a mixture between a
birdish.
cassowary and a spoon, or no, a
Yeah.
shoebill. It looks
A
kind of like a
shoebill.
shoebill.
Yeah. Can you imagine just instead of those forelimbs, it's just like, I have wings.
Yeah, those limbs don't really look like they're going to be doing much anyway, so might as well get wings.
Get me some
Some wings on there.
Yeah, let's see. So let's see what the name hazelae. Does it say what he named it for? I'm getting into this. The bone growth analysis, it was at least 3 years old at the time it died. It was 92 million years ago, middle of the Cretaceous. An intermediate tyrannosauroid linking the earliest and smallest members to the giants. New name, here we go. Derived from the Zuni Native American tribe word Suski, meaning coyote, Tyrannus, meaning king, and hazelae for hazel wolf, who funded a lot of digs in the basin.
There you go, Hazel. You immortalized for
she
your
got
scientific contributions.
Yeah, that's cool.
Mm-hmm.
Coyote. So this is the king coyote. Hazel, the king coyote.
That is pretty darn cool. That's a good animal name. They did good work.
They did. Yeah. And that, it goes into this aspect of paleontology that, it has to get funded somehow. A lot of funding comes from private funders.
Mhm.
And it's this, every once in a while, researchers can give back and say thank you by naming a species after them. The patrons of the paleontological world. I don't think I like rattlesnakes enough to go in the desert and dig for.
That's the one thing.
You just stomp and you're fine. Just stomp every time you walk.
Stomp everywhere. Rattlesnake,
They'll leave you alone.
scorpions, the rattlesnakes.
Scorpions are another
Jaguars.
issue.
And then my skin cracking and burning and
Yeah, that also sucks.
I'll
Also,
look
I'm
like one of the-
weirdly allergic to the desert. So every time I've gone to the desert, my nose just turns into like a running faucet and I lose my voice.
That's interesting.
Sorry.
Yeah. So something about the dry air, I don't know. It's every
Yeah, well,
time.
your normal climate is got
Moist.
some moist.
The San Franciscan goes to the desert.
Yeah.
What?
It's no longer moist. I gotta get out of here.
Yeah, it's when I went to the Mojave to catch reptiles and amphibians in college, I woke up our first day after sleeping there and I thought I had contracted just a terrible cold. I was terribly sick the whole time I was there, and then on the van ride back, about 2 hours inland, went away. It was bizarre. And then it happened to me again. Oh, every time I went to the Dead Sea in Israel and we stayed nearby the Dead Sea.
You're like, I got a case of the deserts.
Yeah, I thought I was sick. And then we went back in to higher altitudes or to kind of a coastal area, and it went away immediately.
That's crazy.
Yeah.
Maybe you just need to take a saline spray with you.
Maybe.
Whenever you go to the desert. Sorry,
Yeah.
Made you yawn.
You did. That means I'm empathetic towards you. That means I love you.
Aww. I love you too. We could just sit here yawning back and forth across the internet.
And Justin didn't yawn, which means he hates both of
Oh
us.
no! You know what? I
wasn't
wasn't
watching.
looking at the video.
He
I was
wasn't
typing. I was
Okay.
typing. Give me another one.
Unlikely
Give me another.
story.
Test me. Try me out right here.
I can't.
Okay.
You just-
What's
Hey,
the
there
word?
you go.
Oh.
Um.
You know what I would love to do? I would love to do get a big panel on here and read the intro to Dr. Seuss's sleep book,
Oh,
where they
God.
say yawn he says yawn a dozen times in the intro. Because I can't it's perfect. But yeah,
I
you
did a
just start
story on yawning, and I felt like we were all yawning constantly. That was many years
I
ago
remember
now,
this. I remember this.
Yeah.
Every time there's a book, there's been, all the kids' books you read to your child. And Blair you wouldn't know this, but Justin.
I read kids' books to other people's children.
Okay. They're, bedtime books. They have, occasionally they've got, these things. Oh, and the truck yawned, and the animal yawned and stretched. And I like to act things out when I'm reading these stories. And so I'll be reading to Kai, and I'd be like, okay, I need to fake a yawn. And then a second into it, I'm like, oh, I'm really yawning.
It's gonna be real.
I really am yawning. That's actually happening now, without fail, on page 3 of The Little Blue Truck, or whatever book it was. I remember.
Oh my God, that's great.
I'm really yawning again. Oh my gosh. Are we talking about the 2020 calendar already?
I'm behind. It's May. I realized, and I went to the art store yesterday. I was like, oh no! And I ran to the art store and I got a bunch of stuff.
Yay. Do you have an idea for this year?
I do, but I don't want to talk about it because it might fail miserably.
I think you say that every year. Okay.
I sure do. And sometimes it does. The year, last year, my first idea was a huge bust. But you'd never know, would you? Because
nope.
I think that's a great idea for a calendar.
A huge bust.
Oh. Speaking of words like that, I got the best present. Blair, you know Heather and JoJo.
Yes.
And
they're your favorite people.
Yes, and they're some of my favorite people as well. And so I get this text from Heather yesterday. Check your mail. Okay, I'll go check my mail. And I got a box, and I wasn't really able to open it until this morning because I had to put Kai. Marshall's out of town, and I had to put Kai to bed, and blah blah blah, all the things. This morning I open it, and it is a mug covered in pictures of birds and their names, but it's a foul language mug.
Oh my God.
So each bird has a, there's the oxpecker, or the great tit or blue-footed boobies or like every bird on
Nice.
it. And you and in the very bottom of the inside of the cup is just a little tit.
Yes. Oh my god.
And I was like,
amazing.
it's
Wait, is
so
it a
fabulous.
three-dimensional
so-
little tit that's actually in?
No, it's just a picture of it.
So-
Yeah.
So this
But
is this is
they're
the
all
thing.
birds, and they're all these cute, wonderful birds. And I've decided that my new life list for birding isn't going to be, to look for all the
See
birds.
foul birds.
I only want foul language birds. That's
Nice.
it. That's all. Those are the ones I'm going. Those are check it off.
So I don't know.
life
Ooh, I
list.
think I found it.
I don't know if either of you experienced this, but I had a cup, milk cup, like a mug, like a coffee mug for a kid that was from but when you drank it, there was a little frog sitting in the bottom of the cup. that
Yeah,
was,
I
like,
had one
looking
with
at-
a hippo in it.
You had a hippo. Somebody else told me they had a bear. Where
I had a
are
bear.
these?
I
Where
had a
are these?
bear.
You had the
yeah.
bear? Okay, what
I had a
happened to
bear.
this? Brilliant idea. Where
I
did the
loved it.
factory burn down and all the molds are gone? How come this is not a
No.
thing?
They exist.
I have not
That's
seen these.
That's a great question though, because I used to love drinking out of my
Yeah,
mug with the
It was
bear
the
in it.
best.
And I would drink like hot chocolate, especially because I'd
Yeah.
drink it down like, oh, it's
I can
coming.
see that.
I
Oh,
can see its
oh my
head. Oh my
God.
gosh.
Yeah.
That was exciting to me.
Yeah. What happened? How come this is
Hold on.
not?
I'm gonna screen share. I'm screen sharing. Look at this. Is that your mug?
It's so cute.
Oh yeah. That is my mug.
It's on Etsy?
Oh, Etsy
Yeah.
remembers everything cool.
Let's see.
That was it. That was oh my goodness. This is bringing back memories. $35. No, okay, never mind. But it's a great memory. Yeah.
Doesn't come for free. Yeah, the one that I had was just all white. It's this one. It's called Hippo Attack. Here we go. Where'd you go? There you
Hippo
go.
attack.
Yeah, here. I'm gonna screen share again. I'm always afraid to Google with my screen share on because you never know.
Yeah, it's risky.
This one. I had that.
Oh, I like that one.
Yeah, it died.
Hmm.
I dropped it from my nerve damaged hand and it shattered on the
Oh
floor.
no.
I know.
You dropped it and then it looked more like that one image in the image that's half open so you can see the hippo on the side.
Yes. That's also how I broke my my like chemistry, my like beaker coffee mug that had a caffeine molecule on it. It was so rad. I bought it at the Science Teachers Association conference. It's very sad.
It is sad.
Dumb
Nerve damage.
I hate it.
Identity 4, that's a hilarious book. He has a book on his office shelf of North American bird species, pictures of different ways to flip people the bird. Oh my gosh, that's hilarious. Oh yeah, animal requests. We should find out what animals we want.
Yes. Any
Can't
animal
see
requests? I can't see the YouTube chat room. Can you see that?
Yeah, I don't think there are many people in there right now, but maybe they have animal requests.
Yeah. Anyone have any animal requests? You can tweet at me too.
Mm-hmm.
So far, this is my plan. The first one I'm gonna tackle is a box turtle. I'm very excited about that. And then I have somebody said savannah monitor, and someone said shark. I've done a couple sharks, so I might do a ray instead or a skate.
Ooh, that would be good.
Yeah.
I like a ray or a skate.
Yes.
That would be awesome.
Might do a wasp.
Paper wasp.
What's the thing I always forget the name of?
Sturgeon. I don't know.
Oxymoxylaka. It sounds like
axolotl?
Thank
An
you.
axolotl. You could do an axolotl. Those are cool.
Yeah.
Ooh, you could do animals with funky names with lots of vowels in them.
Well,
What's
I'm
the
definitely doing some sort of booby.
Then
That
you'd have
is-
to aardvark, I think, has to be in there.
I would love a blue-footed booby.
Or a masked booby. I really like masked
I know.
boobies too.
You could do a kakapo.
Ooh, cockapos are really cool.
I know. Yeah. What's the other bird?
Everyone always wants you
The
to do
South
bats are
no,
hard.
the South American bird that ferments.
The Watson.
Watson. Yes. The Watson.
Watson.
Yeah.
It's also known as the stink bird, Justin. They're fascinating. We have a sculpture of one at my zoo. So I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
Or just suggestions. You
What's
can figure out what you like.
what's the
I could take-
frogmouth bird? What's the
Ooh, a
frog?
pileated woodpecker. That's a good one. What?
Frog. There's like a frogmouth bird.
Yeah.
What is it called? Is it just?
It's called a tawny frogmouth.
The tawny frogmouth. That bird looks like somebody
love those
drew it and
birds.
it came to life.
So that
What-
is the bird that gave me the realization of why I think I like most animals that I like visually. It's because they have a huge mouth. They have a very small mouth to head ratio.
You could do a dung beetle.
Oh yeah.
Identity 4 said there aren't enough beetles represented in the calendars. You're right. You could move into the
Do a
ants.
rhinoceros beetle last year.
Have you done a squirrel yet?
Yes.
When was the squirrel? I don't remember your squirrel. What year was that?
I think that was
Is that the first run?
no, it was in the coloring book calendar. So the second one.
I can't see in black and white, so that's
Ha.
A hyena.
Oh, that's one.
What are other interests? Have you done any of like a hyrax or
Yes. Hyrax or a pica, whatever you want to call it.
pika? Mm-hmm. A marmot.
what's the one that's a
Note to people: don't eat raw marmot. You'll get the bubonic plague and die.
Ooh, a bed bug.
what's the mammal that's like a giant roly-poly with scales? That's always
Pangolin.
That was also in the coloring calendar.
Again, I can't see in black and white, so I missed it.
Oh, no raccoons yet. I could do a raccoon.
Raccoon, lemur.
Yeah, I could do a lemur. That's a good one. I could also do a red panda.
Mm-hmm.
Red pandas, yeah, those are cool.
Red pandas are neat.
They're
I've never done a
they're
giraffe
ridiculously cute.
either.
Ooh, giraffe.
I got square canvases this time.
Ooh,
So we
do
can
a
go
square calendar.
And we can go back to the
Back to
square.
square. Cool.
Yeah.
Let's see. What about what is the let's
So
see if I
email
can
is good. I could do an email. I love email.
yeah, I could do
I'm lacking in the reptile and amphibian department.
oh. Yeah, you've done a
And
couple of snakes and you did a frog or a toad once, right?
Mhm.
There's
Mhm.
yeah. An aardvark.
I do love an aardvark.
They're
An
cute.
oryx. What's your favorite with the stripy butt?
Okapi.
Okapi. Yeah. An okapi.
Always very mammal heavy though. See, that's always the thing.
No. A crocodile.
Oh yeah.
I mean,
although-
we haven't had any cool stories about crocodile behavior recently. What's up with that?
I know.
Yeah,
They were like, we figured it out.
yeah,
Mhm.
that was all we had. You've stopped doing extinct creatures, so
No,
how
I've
about
done
an
one
elephant
every
bird?
year.
Elephant bird.
Elephant bird. I do an extinct creature every year.
Elephant bird.
Hmm.
And I put it on September because it's Species Requiem Day.
I don't think I've ever noticed that. That's like one of those Easter eggs in the calendar. I love
I
that.
spoiled it.
Mhm.
Yeah, so the first year it was the T-Rex.
Ooh, what are
Second
you?
year it was
Mammoth?
the mammoth. Third year it was the dodo. And then last year it was the Dimetrodon.
Elephant bird.
Okay.
The terror bird.
I tried to do a Tasmanian tiger last year and
It
failed.
didn't work out.
It was not good. Let's see, amphibians.
All right.
Chameleon.
You guys can keep going. I have to say goodnight.
You have to.
Okay.
You going to sleep now?
Eventually, but yeah, for sure.
Okay.
Alrighty.
I don't think there's any business. Oh, November, we've been invited to do a podcast here in Portland. They don't have any money to provide transportation. I don't know if that's something.
Where is this
We can
venue?
talk
What is
about.
this venue? What are we talking about?
It's at the Red Lion Inn. It's called Oricon, and it is the Portland, or I guess this Oregon area science fiction and fantasy convention. And they're going to have a podcast room. And we've been
That
invited
sounds
to
very
podcast.
fun.
Yeah.
It could be very fun. It's November, early November, like the 8th to the 10th, I think is the date.
Okay, yeah, let's go.
But
Let's do it.
it's a thing. It's a thing, but we have to figure out if we want to put money into something like that. I'd love to see you guys in Portland again.
Yeah.
I mean, that's the one place we can have a little bit low overhead, I suppose.
Yeah. Also, can we have a merch table?
Oh, that's a good question. Maybe that. Yeah, I'll ask that.
Offset the cost a tiny bit.
Mm-hmm. Yeah, see if we can sell some stuff. And yeah, that's a great idea. I was just thinking we should get I'm gonna order some shirts, but I think I want to order some shirts that are we need an it's all in your head shirt or a hat. We need a design that goes with it's all in your head.
I could get on that. What would you want it to look like?
I don't know.
Do you want a head? And whose head do you want? My question.
An exploding head. It's all in your head.
Let me think about it.
Yeah.
We can doodle a little bit.
I don't know. Yeah, we could doodle too. We'll throw some ideas back and forth.
Yeah. Or somebody's listening and they want to Tony, you want to work on something?
Yeah, we should have a new shirt design. I think it's all a twist. It's all in your head. It could be a fun shirt.
Yeah.
Yeah, right.
Let's just say science is all in your head, and that would just like printed word on a shirt. That'd be decent.
Boring.
I'd wear that shirt.
Host bobbleheads would be awesome also.
Oh my gosh.
I would run around. I would run around like with my bobblehead. I would take it places.
That would be great. How do you have a bobblehead commissioned? I feel like that's a
So
thing.
there is a company in Canada. Not that I looked at this. That you can go in and they do a 3D scan, this little, and then they basically high-end 3D print a bust of your head. And then you could, I guess, put that in.
Ooh, custom bobbleheads for $26 in 7 days. Love it or your money back.
Oh my goodness.
Wholesale custom bobbleheads. There's lots to be done here.
Bobbleheads.
3,000 body styles. Oh my god.
I feel like.
And you get a bobblehead, and you get a bobblehead.
Yeah, I feel like if we were to do this, though, I think if we want to make money at doing this, I would be more inclined to do a some classic and a bunch of ladies of science bobbleheads. That's the thing that would, for science enthusiasts who may not necessarily specifically want a Blair bobblehead on the dashboard of their car, they might go for Mariah Carey or something like this. So I'm just saying, expand it out a little bit.
Yeah.
Yeah.
Interesting.
We could just have sciencey merch that doesn't specifically have to have.
Yeah.
Your beautiful faces are beautiful faces. But they may.
Everybody wants
us.
one.
They may have just met us that day, but they will know. Like they might want Einstein bobblehead in the rear compartment of their car, or on their desk or whatever. That's
Yeah.
just saying.
Yeah, that's true. That's interesting. I still have some originals from last year that I can sell also if we decide what the just buy it now would be for some of those things.
Yeah.
There's that. We could order some Zazzle items in bulk and sell them in person if we wanted.
Is there? We need it. Oh, there's a Mary Curie bobblehead out there already. We could do some other ones though.
Yeah. So anyway, something. But I've still got.
There's no Sylvia Earle bobblehead.
No.
Okay. I like the idea. Bobbleheads for everyone. Yeah. All right. T-shirts, bobbleheads, calendars, things. Everyone have a wonderful week. Is it time to go?
Yeah.
Yeah. Come to Santa Fe.
Say goodnight, Blair.
Good night, Blair. Say goodnight, Justin.
Good night, Justin. Good night, Kiki.
Good night, everyone. Thank you for joining us again for another episode of This Week in Science. Thank you for being in our chat room if you've been chatting or just for hanging out and being a part of the show. Have a wonderful week. I'll be back here. We'll be back here next Wednesday, Friday, twitch.tv/Dr. Kiki for a science conversation. Hope to see you soon. Happy sciencing. Where's the button? There it is.